So today I'm going to talk about some of the work in our lab where I've kind of realized that we are really shortchanging microfluidics for its ability to have impact in the research landscape. So now I'm thinking like of ways that we should really kind of spread the use of microfluidics beyond just people who are traditionally trained in microfluidics. So I'll talk a little bit about that. So to begin with, sorry, does this work? Okay. So just a little quick blurb about myself and University of Toronto. So I'm from the University of Toronto. Here we have our nice city skyline. Here's our campus is actually situated downtown. And our lab, which is on campus in downtown, is situated right in between microbiome and microfluidics. So my research falls in between microbiome and droplet microfluidics research. And this just has to do with my background starting out in microbiology. And I'll talk a little bit more about that later. But it also informs the way that I see microfluidics. So the way I see microfluidics is that it's actually a really powerful enabler for life science research. And I think a lot of people here would also agree. A lot of us do research where we do microfluidics in something else, right? So microfluidics in service of biology. And this is also exemplified in the way that industry is going. You can see that all the really high value companies or high value products are basically microfluidic devices that you use to do something analytical in biology. So example, digital PCR, these machines are super expensive, but the microfluidics is actually quite simple, right? It's a droplet maker and a droplet reinjector. Same with single cell sequencing here. Each one of these is like $9,000 and really it's just a droplet maker. Also in the organ-on-a-chip field, that is also a very profitable area of research. But microfluidics, once again, is actually quite simple. They're just microfluidic channels. And same with diagnostics, where a lot of the most successful diagnostics, these are just capillary wetting paper strips, right? So very simple in terms of microfluidic complexity, but very high in value. So my point is that I think you can unlock a lot of high value in research with relatively simple microfluidics and some other area expertise. And this is kind of reinforced by just me being here, and then I'm constantly getting colleagues coming here and asking to be the microfluidics maker for them, right? They're like, hey, I saw this paper, like, can you make this? Or like, can you make this thing? I'm like, yes, but that's been published in the microfluidics field, so it won't be anything new for me. But it'll be a lot of value for them. But I can't just spend all my day being the service guy, right? So that brings me to a point, which is if microfluidics can add so much value, then why is it so underutilized, right? And so for us, in this group here, most of us are probably traditionally trained in the microfluidics field. Um, and so that's sort of our privilege in a way. So if you think about why that's the case, um, what I think is that if for people who aren't trained in microfluidics and you Google microfluidics and you get pictures like this. So this is microfluidics fabrication facility at University of Toronto, right? It's a clean room, multi-million-dollar instruments. Every time you want to make something, you got to fully suit up, right? And so if I'm some researcher who's not trained in this and I look at this, I'm like, okay, yeah, no, I don't want to do this. Um, and sort of reinforcing that is these microfluidics applications where you have these really expensive machines made for very specific things, and these are very much quite popular. But the reality is that they are priced out for people who are casually wanting to try stuff, right? So here we have a lipid nanoparticle maker. All it really is just syringe pumps and some injection molded chips, very simple chips, in fact. But they sell it for over $100,000, and I have colleagues that are willing to shell out for it because they don't want to go through the hassle of making their own chips, right? If it's like this. But I think the reality of microfluidics can be quite different. So here, I'm showing you some ugly pictures, but this is my lab. This is my clean room in my lab. So we could use the clean room in the craft facility, but we also just use my fume hood where we have a very cheap spin coater and some hot plates. And using this, we can fabricate devices, which I'll show later, that we use for most of our work in our lab. And to run these microfluidic devices, we have these very simple syringe pumps. They're like $500 each, just an inverted microscope and an industrial camera. So the point I'm trying to make is that microfluidics doesn't have to be that fancy, right? And you can still do a lot of really useful work with it. So you really don't need $1 million equipment to make useful microfluidic devices. And so I want to make the distinction then between microfluidics in terms of power or advanced microfluidics or casual microfluidics. And this already exists in biology. So you can have super fancy microscopes where I would not want to touch this microscope um, because you need a lot of training on it, and not everyone has access to this, right? But then also every biology lab has these small casual microscopes. I wonder if you can see my pointer. Uh, okay, regardless.
We can see
Yeah.
it.
So, okay. So every lab has these casual microscopes, and these things are actually really powerful. Like it enables a lot of science. And so there's no barrier to accessing these, right? They're cheap. It's really hard to break them, and you give like a 5 minute introduction, and you know how to use it. And so the question then is, can we do the same with microfluidics, right? Everyone knows how to use a simple microscope, but can we do the same for simple microfluidics and to add value to biological research? And so in service of that, I'm going to talk through, okay, now I gotta clear this, my bad. Okay, so I'm gonna go through my own personal journey in microfluidic accessibility, and I know that probably many of you have had similar thoughts. So my background is in microbes, and microbes turns out are highly heterogeneous. So even if you take the same culture of pure microbes, you'll find that between the different cells, there's a lot of differences. And back when I was in training, I realized that nobody is able to address these differences because we just don't have the tools. So that's why I went to train with Adam Abate, because back then I realized that with droplet microfluidics, we can tackle this problem. So here we have um, a schematic of single cell analysis in droplets where we can just put different bacteria into different droplets and then do single cell analysis on them. So I spent my time developing a lot of workflows using this droplet microfluidics. And then I ran into the problem where it turns out that nobody wanted to use these workflows because everyone wanted these simple one button casual uh, workflows, and that totally makes sense, right? So now we realize that microfluidics, if you want it to be highly used, then it can't be the power user microfluidic. It's got to be for casual users. And so now I'm just going to walk through some of these examples without going into much detail. So if I wanted to do single cell sequencing, so starting with a bunch of different cells, we can encapsulate them into droplets where we get one cell per droplet. And through a bunch of microfluidics operations, we can add different reagents. So we can lyse the cells, fragment the genome, and we add unique barcodes. And so these unique barcodes can tag the DNA in each droplet. And so when we finally do the sequencing of all the pieces of DNA together, based on these unique DNA barcode tags, we can pull out the sequences that belong to each droplet, which basically is belonging to each cell. And by doing this, you can get a genome database of whatever your sample, where all the genomes are separated out based on the individual microbe, and you can cluster those microbes into their species to do high resolution analyses. So without going into the detail, I can tell you that these devices are very complicated. So I published kind of two different variants of these things. And one requires 3 devices, one requires 4 devices. And here's one of the devices, quite complicated, right? You need to make droplets, and then you need to merge them, pair them with barcode droplets, which you make on another droplet maker, and then you merge them, and then you pair them on the same device with another droplet, and then you merge them again. So just looking at this, you can tell that this isn't going to be something that's easily picked up by anybody. And so many years later, actually, another group made a similar workflow, but with 5 devices. So you can imagine that with something like this, you're going to need really high quality devices and expertise in handling, right? Because if any of these steps goes wrong, and then your whole workflow is ruined. So these are more like concept cars rather than practical things that people use in their daily lives. And so realizing that, I decided to start tackling how do we simplify these microfluidics workflows. So to that end, I started combining the biology and the microfluidics. And what we came up with is what I call droplet targeted amplification sequencing, or DOTA-seq for short. So in this workflow, it's much simpler. You start off by encapsulating single cells in droplets as before, and then you turn them into a hydrogel. So this is porous, allowing reagents to diffuse into the hydrogel. And that way we can do multiple steps without doing microfluidic steps because you're just diffusing things in and out. So that makes it easier. So with this method, you can lyse the bacteria, and then you get your genomes in these hydrogels. And then you put it through another microfluidic droplet maker. And this time you put in some PCR mix with barcode molecules. So these are single molecule barcodes, but each of these are unique. So in these new droplets, you thermocycle them for PCR, and then you have primers that amplify the barcodes and they amplify your genes of interest. And then because of the overlap on these primers, they can make a complete DNA fragment that contains whatever you want to sequence as well as a unique barcode. So doing this, we're able to take the previous 4-step workflow into now a 2-step workflow with very simple devices, right? These are just droplet makers. So as a result, this has become the daily driver of our lab. Finally able to get biology students to do it without being scared. And so this kind of really shows the importance of taking complex workflows and making them simpler. And it really does add value to the actual work. So having done that, though, I still run into another problem, which is that collaborators still don't want to use droplet makers because there's no commercial solution, or the commercial solutions are complicated, and they don't want to go into the clean room, right? And so even for my students, it takes a full day to fabricate a new design in a clean room. And so we started thinking, okay, how can we make this to shorten this cycle and make this more accessible? So the major instruments that we use are a spin coater, which is quite cheap, but also a mask writer and a mask aligner. And these are multi-million-dollar instruments. So the idea is, can we somehow make an accessible alternative to these? So what we came up with is actually to use an LCD screen as a digital mask. So the way photolithography works is you have a light source and then you have a mask, right? And that gives you the patterning on your photoresist on your wafer. But if you replace the physical mask with an LCD screen and then you just program the pixels to let light through in the pattern of your device, then you basically have a digital mask. And now you can directly translate your design to your mask very, very quickly. And it turns out that this setup is actually very cheap, and we don't even have to build it. And the reason is 3D printers. That's how resin 3D printers work. So here I'm showing you an example of a 3D printer in our lab, which we reprogrammed to basically project a microfluidic mask. And you can see that here you can just see the mask, and we just stick our wafer on top of it, and we can get the devices fabricated. So how well does this work? We started printing these calibration slides to kind of get at what kind of features you can get. And so here's a mask, and then here is our printed wafer, for example. And here I can show you a zoom in where here we're looking at holes. So we print this line and then we try to make holes in there and see how small we can get the holes to be. So if you quantify all this data, what we find is that if you're looking at basically if you have thinner devices, then you can get smaller features. So we choose 40 microns to characterize here because our devices in our lab are typically 40 microns high. But if you try to make a channel, you can see that if you try to make them too small, they actually start plateauing down at around 100 microns. But with some tricks, actually, we can make them even smaller. But anyway, you can see that there is a trend, and you can make a calibration curve where you have a reference size that you draw on your CAD and the actual width that you get in your device. And so it's not perfectly one-to-one, and it gets to be about one-to-one after about 500 microns. And it's because of the pixels. The pixels aren't perfectly collimated, right? So the light kind of shines to the side. And we can actually improve this with further designs down the road. But regardless, using this, we can now predictably draw a design on our chip and then fabricate it and get the expected channel width. And the same with pillars. So here you can see we can make about a 60 micron pillar, and then that can go all the way up, where it becomes basically one-to-one later on. And same with holes, right? You can also make holes, and you can also make about a 50 micron hole all the way up, and it becomes predictable as you make them bigger and bigger. So having done that, we also decided that, well, what if we wanted to make multi-layer devices? So we came up with this physical alignment manifold where we basically 3D printed this manifold that it's stuck on the 3D on the LCD screen. And that allows us to basically take a piece of the manifold and then put in slides so that it fits only in one orientation and that we can physically squeeze it into the corner. So it's in the same place every time. And we at this point, we started using glass slides because it's even cheaper than silicon wafers. And so with this thing, you can kind of deterministically fit everything into one spot and then put them back on your LCD screen so that everything goes back the same way. And then if you twist the knob and make sure everything is snug, then you can do your second layer of exposure, and that does the alignment for you. And so here's what this looks like. And all of this done is done with the exact same 3D printer we used to fabricate the devices. And so now if you look at it, here I'm showing you an example of two-layer device. Here's our alignment marks. In the blue is the profilometer. So in blue, you have the first layer or the wafer itself. And then on the green, you have the second layer. And then red, you have the third layer. And you can see that there's the alignment isn't perfect. But if you quantify it, you can see that we're getting at about close to 50 microns offset every time. So actually, this is good enough for most of the devices that I need to make in my lab. And so this has become our daily driver of device making as well. So here I'm just showing you some examples of droplet makers that we can make with this. It's making perfectly fine droplets. And also we can also make lipid nanoparticle generators. These are very simple devices. And we're able to generate lipid nanoparticles reproducibly. And so these are replacing really expensive instruments, right? And so the idea is because they're so cheap, then every lab can have one, right? And you don't have to have access to $1 million clean room to now fabricate your own devices. All you need is wafers, which if you use glass wafer, it's super cheap. And you can have spin coaters and just buy a printer, which is about $500, print your own alignment platform. And now we're reliably going from design to device in basically 3 hours in our lab. And so this has really sped up the rate that we're iterating on new ideas. Now, if I have a new idea, I'll just try it. Before, we had to go print our masks, and sometimes it took a long time, and it kind of was a barrier to trying random things. So hopefully, with this now, labs that weren't traditionally trained in microfluidics don't feel like it is a huge barrier to try it. And some random grad student could buy these components and just start messing around in their lab. Okay, so to wrap up, I want to make the point that I think casual microfluidics is something that is not really exploited enough in the research landscape. And simple microfluidics can have really a high value, right? And if we can now fabricate these devices on the benchtop, then I think there is a lot that we can do that's unlocked before, whereas previously my colleagues might be like, oh, I can't buy an instrument for this, or I don't want to invest in this. And now it's possible. And I know that there's already a lot of exciting work going on in microfluidics accessibility from the community. So I think maybe right now we're at a time point in the field where we can start pushing for casual microfluidics, where we can really have the impact of microfluidics go in beyond just the labs that are traditionally trained in it. So can we make microfluidics as ubiquitous as microscopes? I think that would be really cool if we can. So with that, I just like to wrap up. So here's my lab as of a year ago, and this is Tian. He's a student. He's about to graduate his master's. The 3D printer work is done by Tian, and I'd like to thank my funding. And yeah, I guess that's about it.